Pelle Redare, are you ready?
I am.
To present iCellate and reinventing cancer management. Welcome.
Thank you. I'm impressed to see so many of you here today since we have a fierce competition with the weather. So thanks for joining us. It is well known that cancer today is in general detected quite late in the disease progression, and some patients get some harsh treatments as well. However, we already have some very interesting technologies available that has the potential to make a total game changer in the market. So these techniques could be used for early detection, for monitoring of the disease, and it's also extremely intriguing to see how it can be guided for targeted therapies. So, starting with the tumor cells, the circulating tumor cells which spreads the disease itself. It has been known for more than 150 years that the primary tumor detach circulating tumor cells that spreads either into the bloodstream or the lymphatic system. Most of these small cells commit suicide, but some of them will be viable and migrate out and form metastases. As I said, this has been known for many, many years, although it is quite recently that we have been able to isolate them, since especially in the beginning of the disease, they are at extremely low concentration. So just a few cells among tens of millions of other cells. So our technology is divided into 3 steps. So the first one, we need an ordinary blood draw. Then we take that blood draw, put into our instrument and isolate the cells. And then when we get single cell resolution, we put them into a sequencing instrument in order to get the genetic information. So our isolation instrument looks like this, and we put the test tube in the middle of the machine. We push a button, and then it takes approximately half an hour, and the machine automatically isolates the tumor cells. We have a non-marker dependent technology, so we isolate them on biomechanical characteristics. So this means that we are able to catch them even when the cells are in their most malignant phase. We have a patent granted in China. It's still pending in the US, Europe, and Japan. And we are waiting a final verdict from the authorities in Europe and the US here in the summer. So after we have isolated them and we get them cell by cell, we can send them for sequencing. This is a project we are running right now. So we are so close to the border to show that we can sequence them as well with our technology. And this is important since that confirms that we really have caught cancer cells. It is a nightmare, of course, if you get a sample from a patient and you tell that patient that you have cancer and that shows to be wrong. But with genome sequencing, we will get rid of the false positives. So that's important even in the early steps. If we look further down the road, it also opens up tremendous opportunities when it comes to personalized cancer medicine. So what have we done so far? Our instrument is very well verified. In our technical file, we have 571 samples. So we took 571 tubes with human blood, and in each tube we spiked in 5 or 10 cancer cells. And then we ran that through our machine, and we were able to detect cancer cells in each and every of the 571 samples. We did that with a high recovery rate of almost 90%, with a very small spread as well. So that tells us that the method is robust and stable and works. We have performed, or we are actually 10 patients short to include 3,500 subjectively healthy patients. So they arrive at the hospital for some other reason than cancer. They have no history of cancer before, but they are asked if they would like to be involved in a screening study. They have to belong to a high-risk group and be in the age of 45 to 80 years old. So far, we have analyzed 3,027 patients, and we found 89 positive samples, which corresponds to 2.9%, which in this high-risk group with this age is fully in line with what the literature suggests. So, business-wise, we will start with screening. We have an ongoing dialogue with BNP Paribas, one of Europe's largest health insurance providers. Their customers are asking for a service to be screened yearly for cancer. And further down the line, we are looking into exploring monitoring and personalized cancer medicine as well. So we are in a financing round right now. We are asking for 15 million Swedish at a valuation of 30. We expect to close this round in the beginning of June, and we have got 5 million of this 15 on the bank account already. And we have met a lot of larger players that tend to invest later in the process. So they have asked us to fulfill a couple of milestones, and then they are ready for a larger round in 2018. So if you are interested and would like to buy at a higher price, you have a second chance as well. Thank you very much.